see.
All right.
So when the light goes green, you can start.
Okay, thank you, Brian, for the introduction. Thanks to the organisers and all the societies supporting the meeting for the invitation to come and talk on what's become known as Fisher's infinitesimal model. This talk, I'm sorry to disappoint you, but I won't have much maths or hardly any equations. I'll really be trying to give an overview of this, as you'll see, somewhat mysterious thing, the infinitesimal model. And it's worked jointly with Alison Etheridge and Amandine Weber, and it's based on a paper we published in TPB, Theoretical Population Biology, last year. We were inspired to write this really when I read a draft chapter from Walton Lynch's magnum opus, a massive book which I strongly recommend for weight training as well as for intellectual content. So Bruce had, I think, written this chapter on the infinitesimal model, and I read it and exchanged views with him. And basically, it bore no relation to what I thought the infinitesimal model was. And then it emerged that really, although it's talked about a great deal, it's something that really has very murky origins and is quite hard to define, both in terms of what the model is precisely and what is its scope of application. So we wrote this article which attempted to really pin down the model in a bit more precisely. And alongside the paper in TPB, Michael Torelli wrote a commentary in which he pointed out that actually, indeed, it is used in many different senses. The infinitesimal model means many things to many people. In the broadest sense, it refers just to the idea that complex traits, as we now call them, continuously varying traits, are influenced by very many genes of small effect, and that's just a general view of the genetic basis of complex traits. The sense in which I'll use it is one that can be, I think, justified mathematically, given certain assumptions, and that is one in which we think of the pedigree, a set of individuals with known relationships. We think of some ancestral population, in the simplest case, which is well mixed, and of genes percolating down through this pedigree and influencing the traits carried by the various individuals. And the crucial core of the infinitesimal model is that the joint distribution of trait values of all these individuals is Gaussian. It's a multivariate Gaussian if there are multiple traits. And it's a multivariate Gaussian with multiple individuals, possibly one trait, possibly more traits. And so Torelli calls this the Gaussian descendants model, because it implies that regardless of the trait values of the parents, if we condition on the values of two parents, the distribution of the offspring will be Gaussian with a variance independent of the parents' trait values, and that's the crucial point. The multivariate normality of the whole distribution of all the individuals ensures that if we condition on certain individuals, that is, if we select, then we'll alter the means, but we won't alter the variances and covariances. So that's what I think of as the core of the infinitesimal model. But Torelli points out that actually the term is used possibly more widely to refer to a stronger assumption, which is really only an approximation and really can't be justified, certainly in the presence of selection, and that is that the whole population follows a Gaussian distribution. And that he refers to as the sort of Gaussian population model. So what I think of as the classic infinitesimal model, what Torelli calls the Gaussian descendants model, is actually very broad. As I've tried to explain, it includes selection, because by conditioning on the values of any set of individuals, we don't alter the covariance of their descendants. It includes recombination, at least amongst unlinked genes. It can be extended to include mutation. It, of course, includes random drift in the sense that if we can specify a process that generates random pedigrees, and even with a fixed pedigree, there will be random segregation generating random fluctuations. It also includes population structure, gene flow, and so on, because we can have any pedigree we like. We can have a pedigree that consists of subpopulations, has non-random mating, etc., etc. But I think what I want to emphasize in this talk is that the model really doesn't require that traits are additive. And I think this is important because I've often felt that molecular biologists poke fun at quantitative genetics. They say, oh, you're just dealing with additive models, you're dealing with weightless elephants on frictionless surfaces. And of course, the world isn't additive. It's an absurd assumption. And so we really do need to understand why it is that the infinitesimal model and the whole framework of quantitative genetics actually works rather well, even in a highly complicated and highly nonlinear world. Another point to emphasize is that it doesn't imply that allele frequencies change infinitesimally. And this is an especially important point to make in the world of evolve and resequence experiments, where we follow frequencies of individual SNPs, which will change up and down and fluctuate. It may change very greatly in a small population simply through random drift. The point about the infinitesimal model is not that allele frequencies don't change very much, it's that the distribution of allele frequencies is hardly perturbed by the action of selection. And that's a subtler way of looking at it, I think. Okay. So I'll give a sort of very rough and very crude history, and I'm slightly, I realise there are many people in this room who know much more about the history than I do. This is my attempt to summarise it. I think that the idea actually behind the infinitesimal model goes way back to the mid-19th century, to concerns about blending inheritance, criticism of Darwin's theory of natural selection made, for example, by Fleming-Jenkin in a review of The Origin of Species in 1867, in which he pointed out that if inheritance was a process of blending together the properties of the parents, then the variability in the population would be blended away to produce a kind of grey sludge with very little variability. This was actually an argument quantified in one of the very earliest quantitative calculations in population biology by Davis, 1871, who showed that the variance, as Fisher termed it, would decrease by a factor of 2 every generation under blending inheritance. Galton, of course, was deeply concerned with inheritance and developed, or at least inspired, many of the statistical methods that are necessary to understand inheritance of quantitative traits, founded biometry, really. And he observed that the variance amongst offspring would be surprisingly independent of the values of the parents. There's a very nice quote from a paper in Nature, 1877, where he was describing the results of some crosses between sweet peas that he made. And he said, I was certainly astonished to find the family variability of the produce of the little seeds to be equal to that of the big ones. But so it was, and I thankfully accept the fact. For if it had been otherwise, I cannot imagine from theoretical considerations how the problem could be solved. And that indeed is correct. It's this radical simplification that we can treat the variances and covariances amongst offspring as independent of the parent trait values that makes the infinitesimal model so powerful. And I actually wish more papers were written like this in Galtonian prose, but anyway. Okay, so Galton tried to develop what he called a law of ancestral heredity, in which he tried to suppose there might be particles which have a certain probability of being expressed in successive generations. There's an interesting article by Michael Bulmer which explains the relationship between this model and our current understanding of quantitative genetics. Carl Pearson, around the turn of the 20th century, formalized Galton's observations, developed the statistical machinery, much of it that we use now. And famously, Fisher in 1918 showed that Galton's observations are actually consistent with Mendelian genetics, provided we assume a large number of freely recombining genes with small effect. So the infinitesimal model is attributed to Fisher. It's Fisher's infinitesimal model. It's actually slightly frustrating to read Fisher's 1918 paper and try and find the model in there. He never uses the term infinitesimal. And it seems the term infinitesimal was used by Michael Bulmer in the around 1970, and Bulmer says that this model is widely called the infinitesimal model, but no one seems to remember where the term came from. And so I asked Michael Bulmer, and he said, well, he didn't remember, but maybe Bill Hill remembered, and then Bill didn't remember. So it's rather alarming that the origin of the model, of the term anyway, is kind of lost in the mists of time. even in 100 years. Anyway, so following these developments, quantitative genetics, I've said it developed in obscurity. This is possibly not quite the right term to use, but what I mean by that is that there was a very sophisticated machinery developed primarily for animal breeding and plant breeding, but this developed largely independently from evolutionary biology. At least, if you like, it wasn't applied to outdoor problems in natural populations until really the 1960s, 1970s, when work by Alan Robertson, Russ Landley, Michael Bulmer really applied the quantitative genetic methodology to evolutionary questions. And I'll say some more in a minute about a really nice paper by Alan Robertson in 1960, which sets a very robust limit to the change in the mean, that is the response to selection that one can have, based solely on standing variation in a population of a certain size. And understanding Robertson's result here actually tells us something about the relationship between the quantitative genetic view, looking at continuous traits, the biometric view, and the population genetic view. So I've talked about the infinitesimal model and talked a bit about the history, but I haven't actually said what it is. And as Torelli's commentary suggests, there actually are a series of sort of nested versions of the model. I'll try and define it first purely at the phenotypic level, without referring as far as possible to genes as such. And I'll neglect non-genetic variants. That's a fairly trivial thing to add in. I'll assume for the moment additivity. I'll assume we're dealing with haploids, again dealing with diploids, it's essentially the same. And the core of the model is that if we take two unrelated parents with different trait values, Z1, Z2, their offspring, the genetic value of their offspring, the breeding value, will be distributed as a Gaussian centered on the mid-parent value and with a constant variance independent of the parental traits. And I've called that so-called segregation variance V0 over 2, and clearly with random mating, with no selection, if the population is just left to reproduce under this model, it'll very rapidly converge to a distribution in the population with a variance V0, and that'll be a Gaussian distribution. So the one elaboration which one needs to fully define the classic infinitesimal model is to say what happens when the parents are related. In a finite population, parents will become related inevitably, and then the variance amongst offspring is simply reduced by a factor 1 minus f, where f is the probability of identity by descent, the probability that two genes coming together in meiosis will be identical by descent from some reference ancestral population. And this term, this effective relationship, occurs simply because the segregation variance here, this V0, is due to segregation of alleles in meiosis producing the next generation of haploid offspring. And it's this generation of variance, generation of genetic variance through recombination, through segregation, that offsets the dissipation of variance by blending inheritance that concern Fleming-Jenkin and so on. So it's the introduction of this term that basically saves natural selection, that preserves variation, and it's the constant value of this term, which means that variation will be preserved for a long period of time, limited in the core model by population size. And one can add in the effects of mutation in a slightly ad hoc way, assuming some kind of house of cards model of mutation, by simply saying that the segregation variance is augmented by some value, some constant value, due to mutation in the underlying genes. But I won't say more about that. Okay, so this infinitesimal model defined at the phenotypic level appears to be completely inconsistent with Mendelian genetics, and maybe that was the concern of these early reviewers, because offspring from extreme phenotypes must have low, or in fact zero, variance. If we imagine just a whole range of genotypes which produce various trait values, the genotype with the smallest value will be, in general, a unique genotype, and so offspring from that extreme phenotype will have no genetic variance. Actually, a cross between the smallest and the largest will have no genetic variance. There'll be an F1 which will be entirely heterozygous. The F1 will be completely intermediate or whatever. I'm not saying anything here about additivity or whatever. I'm simply saying that in general, extreme phenotypes must have a low variance, regardless of the mode of inheritance, almost. So the crucial assumption is that the actual phenotypes in a population occupy a very narrow range relative to what's possible. So we have this huge range of genetic possibilities, but in a real population, a typical situation, we'll have a very narrow range of variability. And a consequence of that is that each phenotype corresponds to many, many different possible genotypes, and therefore specifying, conditioning on the phenotypic value tells us very little about the genotypic value at any particular locus. If we know the phenotype, we actually don't know the genotype in almost all circumstances. And so it's this assumption that is crucial, and it's a very well justified assumption, because we know that if we take a population, pretty much any population, any genetically variable population, and we select up or we select down, we'll get a response of many, many standard deviations. We know, in fact, that the extremes of what is possible, even based on standard variation, are way beyond what is actually realized in almost all populations. Okay. So I'll turn now to go through in detail Robertson's results, which set out a limit to the change in mean one could achieve selecting for an indefinitely long time on a certain pool of standard variation. And I'll do this in order to try and connect the phenotypic level that I've described, the phenotypic version of the model, to the underlying genetics and see what it's assuming about the genetic basis of the traits. So one can derive Robertson's limit in an almost trivial way by saying that the change in mean in one generation is equal to the selection gradient beta times the additive variance in that generation. Therefore, the total change over the whole span of your selection experiment is the sum proportional to the sum of the genetic variances. And if the genetic variances decrease by some factor every generation, factor 1 minus 1 over n for haploids, 1 over 2n for diploids, then summing that geometric series, we just say that the total change in mean is equal to the change in the first generation, beta times VA naught, times n. And it's simply saying that because genetic variance in a population of size n dissipates over timescale n for haploids, 2n for diploids, the total change is simply population size times change in the first generation. So that seems very simple. It actually can generalize to include epistasis, because we actually know how the various different additive by additive components of variance, etc., dissipate in a finite population, and we can make the same argument. And we actually find that with haploids, with arbitrary epistasis, we find that this is simply the same result, but this is now the total genotypic variance. That is the sum of all the initial variance components. In diploids, the additive by additive contributes twice as much, additive by additive, 4 times as much, and so on. And we try and ignore the nightmare of dominance. So that's a bit of an aside. Going back to Robertson, Robertson actually derived the result not by this simple argument, but by looking at the level of individual genes and pointing out that the total change in the mean, the expected change in the mean, would equal the sum of the effects of each gene times the difference between the probability that that allele would be fixed in the population minus its initial frequency. If there were no selection, these would be equal. There'd be no expected change in the mean. The expected change overall depends on the excess of fixation probability over initial frequency, which is generated by selection. And so if he substitutes in Kimura's formula for the fixation probability, and then assumes weak selection, assumes that the product of population size and selection per allele is small, then you get this formula, you substitute in, and you recover Robertson's formula. And so what this shows is that the infinitesimal model is really assuming weak selection at each locus. It's assuming that selection is weak relative to drift on each of the underlying alleles. And in fact, the success of Robertson's formula, there's a nice survey a while ago by Weber and Diggins, which has surveyed many, many selection experiments on many organisms, and showed that actually Robertson's selection limit works pretty well. Very well actually for biology, I think. And that implies that selection cannot be strong enough to significantly accelerate the loss of variability in the population. So that's so far the infinitesimal model defined at the phenotypic level, and I've tried to explain the connection using Robertson's argument with the population genetics at the underlying allelic level. but the question is, how far does it generalize? Well, the key idea is that the joint distribution of individuals on a pedigree must be Gaussian. And if it is Gaussian, then conditioning on some individuals, that is selecting on the population, does not alter the covariance. So really what we're asking is, under what circumstances do we get a multivariate Gaussian? And that's really asking about the central limit theorem. And so the paper we wrote was about applying various versions of the central limit theorem to show that we get this result not just with additive models, but also with certain epistatic models, as long as there's in some sense not too much epistasis. And what's still an open question is just how much is too much. Okay. One can clearly come up with counterexamples to the infinitesimal. If we take a trait that follows the infinitesimal and we square it, or we apply some scale transformation, then we'll generate something which is non-Gaussian. So there we seem to have a clear counterexample. That is because that kind of scale transformation introduces systematic epistasis pointing all in the same direction defined by the scale transformation, and that violates the conditions under which we can derive this multivariate Gaussian. So actually the question that we're concerned with is not so much developing the mathematical machinery, but trying to understand why it is that real organisms with real systems of gene regulation and so on, why those should seem to live in the world of the infinitesimal model. So I'll just sort of finish up by giving a sort of concrete illustration, just illustrate with some simulations. And what I'm doing here is simulating initially 1,000 loci, n is the number of loci, small population of 100 haploid individuals. The top row here is the additive version, where additive effects are of order 1 over square root m, so that the additive variance is of order 1. The additive variance starts out at 0.13, which is an arbitrary value. The dashed lines are the neutral case here, the solid line, the selection, strong selection here, and you get a selection response. Not surprisingly, the selection response slows down as additive variance is dissipated. The additive variance is almost unaffected. In fact, you can't see the difference on this scale. It's almost unaffected by selection here. If we look at the model where we add sparse pairwise epistasis with effects of order 1 over m, so that the additive by additive variance is of the same order as the additive variance, then we get these results. We again get selection response here. And here we have the additive variance, which is much greater now because, as is well known, the main effect of epistasis is actually to generate additive effects, because epistatic interactions will have marginal effects that feed into the additive variance unless the allele frequencies are rigged in a very special way. There's also a substantial additive by additive component that I put in this model. For the first 30 or 40 generations, there's not much difference between neutral and selected. In other words, the infinitesimal model is working pretty well. It starts to break down here for the additive component, but not the additive by additive, which still works pretty well. So then we can sort of extend this and say, how does the deviation between the selected and the neutral case, that is, how much does selection alter the additive variance as we change the number of loci? So I'm expressing that by taking the initial gradient through time, the rate of decrease of additive variance, or log additive variance, that's the initial slope, times n, which for the neutral case, these small dots, is expected to be 1, indeed it is on average. And we're asking, how much does selection cause a perturbation? So these dots are additive equal effects, and so we get a convergence with 1,000 loci, pretty close agreement. We saw that before. With these grey dots, we have exponentially distributed effects. That actually reduces the effective number of loci, really, so we get a bigger deviation. And crucially, the large dots are where we put in this model of sparse pairwise epistasis, and we get a scaling of order 1 over the number of loci. As the number of loci increases, we approach this infinitesimal model limit. So, in fact, there's also an open question as to why it works actually in the simulations rather well. 1 over m convergence. In the maths, we've only got a kind of a limit of 1 over square root m, which is a weaker result, but it applies more generally. So there's also a question of under what circumstances do we get this fairly good agreement with the infinitesimal? Okay. So I'm going to wrap up here and because I hope there'll be a vigorous, although perhaps not too vigorous, discussion afterwards. Okay. So what I'm trying to say is that we have an infinitesimal model which can be defined at the level of the phenotype. I defined it for the additive case, and then I sort of outlined the extension to the non-additive case. And the basic idea for the non-additive case is that we can take our understanding from classical quantitative genetics, initiated by Fisher in 1918, which actually tells us how not just the additive component of phenotype changes and what its distribution is, but how the additive by additive component, the dominance component, all of these components of phenotype, we know how those change in the absence of selection. They change basically as a linear function of the variance components and the identity coefficients. It's kind of a bit of a nightmare when you get up to higher order variance components because you have to get higher order identities, but in principle it's a straightforward idea. We have this neutral model of how the genetic covariances change, even with non-additivity. And the idea is that if the whole distribution is multivariate normal, then that covariance structure changing through time will be unaffected or only weakly affected by selection, and therefore we can use the machinery of classical quantitative genetics, including dominance, including epistasis, to describe the evolution of the population even under selection. So we have a model here which includes selection, drift, mutation, recombination, gene flow. I'm being a little bit slippery here because so far I've assumed no linkage, and Himani Sachdeva this afternoon will talk about the extension of the infinitesimal model to include linkage, which makes quite a different and even more interesting kind of a beast. Anyway, so there's the model. There are plenty of open questions. There are technical questions about exactly what assumptions are compatible with this sort of central limit theorem, multivariate normality. When do we expect to see something that looks, as we actually see in real data, that looks infinitesimal? But there's also, to me, as primarily an evolutionary biologist, this question of, does it help us understand long-term evolution? Now, in some sense, it really can't, because the prediction would be that the genetic variance for any trait would be determined by a mutation drift balance, and it would equal population size times Bm. And that would imply a very strong dependence, a linear relationship between population size and additive variance, and we don't really see that. Of course, this is exactly the same puzzle as Lorentz paradox, that at the molecular level, sequence diversity doesn't depend linearly on population size, it has a much weaker dependence, and I think that still is not fully understood. There are many competing explanations for why that's the case. Maybe these questions are quite related. And so, in fact, understanding whether long-term evolution can be reasonably described using the infinitesimal model, which is very attractive because it's a very simple model, that boils down to asking whether the underlying genetic variation is selected rather weakly relative to drift. What is NS typically for the alleles responsible for adaptation? And more broadly, what shapes the genetic variance? Why is it that we see heritable variation at broadly similar levels across such a wide range of populations? So I'll finish, though, by maybe, hopefully rather provocatively saying, well, if we really think the world is more or less infinitesimal, if we can actually explain the patterns of inheritance and evolution of quantitative traits using the infinitesimal model, then that implies that the underlying genes are really very large in number and have very small effects. And this is the kind of argument that Boyle et al., Jonathan Pritchard and others made in a paper in Cell last year, where they argued that actually there may be an enormous number of genes, and even more provocatively argued that these genes affecting traits may not be concentrated in particular pathways. They may be spread over the genome over many, many different biological pathways. So then you ask, well, why bother finding the genes? Why not just live in statistical heaven? That's my argument. Okay, thank you.
I didn't get to use my traffic lights.
I just beat you to it.
Well, thanks for a great talk, Nick. Are there questions? The helpers, could you grab some of that hand mic so you can speak?
Thanks. I was surprised by your summary that Robinson's limit works well, because it says that the limit is reached because all genetic variation has been lost through drift. And virtually no selection experiments show that unless they're very tiny populations.
Yes. So I should have been clear that I was actually referring really to the Weber and Diggins survey, which actually didn't give Robinson's limit, which, as you say, wouldn't ever be reached. You always had mutation at that point. They looked at experiments at 50 generations, and they used the analogue of Robinson, so it's summing the sum up to 50 generations and comparing it with the observation, and that works pretty well. And they also did consider mutation, and they put in what estimates they had of VM, and showed that by 50 generations it shouldn't make very much difference. If you go to 100, 200 generations, then clearly it will. So I'm not implying that selection response is based on standing variation, but rather than that the infinitesimal model, actually plus or minus mutation, will give a good prediction, even in very, very varied traits and organisms.
Doesn't a model of small effects inevitably apply in the end additivity?
You think of a kind of Taylor series argument that you could make. Yes. It's just not clear to me that you can make a Taylor series at the level of individual genes. So clearly, if you had some enzyme and you had small variations that were changing it very slightly in the range of possibilities, that would be the case. But it may be that there are alleles with small effects on the trait that have a large effect at the genetic level. Maybe, I mean, if it's true that they actually have small effects at the genetic level, at the flux level and so on, then you can make the Taylor series argument.
That's what I'm talking about.
Yeah.
Hello. Oh, hello. So one potential answer to your provocative question is that so when one does a GWAS survey for finding genes underlying a trait, one tends to find, say, a handful of major effect genes, which would not be covered, as I understand it, in the infinitesimal model, and then a bunch of kind of minor effect genes that only weakly contribute to a trait, which can be more accurately captured by an infinitesimal model. So I was wondering, has there been any theoretical intuition about how maybe the effect of rare large effect genes could kind of somehow skew the underlying distribution of additive variance?
Yeah. So clearly there's a range of effects. There's a distribution, and let's say it's exponentially, it's a very skewed distribution. There's some big ones and lots of small ones. But on the whole, the large effect genes don't have a large enough effect to substantially alter the change of variance, which does fit overall the sort of infinitesimal prediction. But to me, I mean, the way to phrase it would be to say, if we look at some long-term adaptive process, to what extent are the large effect genes contributing versus the small effect ones? Initially, as we see actually when we make a major challenge with insecticide spraying or whatever, you do get large effect genes coming in. But it may be that those over time will be ameliorated or augmented so that at the end of the day you see actually a much more mixed picture. But yeah, I don't want to say that I believe in the infinitesimal world. It's a beautiful thing. It would be wonderful if we lived there, but but of course it's a mixture. And the puzzle is to understand why quantitative genetics works so well, even given that things are not completely infinitesimal.
Recently, Miguel Ángel Toro made a comment in the Journal of Animal Breeding, commenting that when you apply the infinitesimal way of working with dominants, and you take small bits of dominants, infinitesimal parts, you arrive to a paradox. You have to increase the consanguinity until infinite, or if you keep the consanguinity, you cannot keep, this kind of infinitesimal model for dominants. I don't know whether you have read about this. It was quite recent in the Journal of Animal Breeding and Genetics, and it looks like a kind of, uncomfortable flaw in a nice theory of infinity.
So it's clear that it all breaks down when you have too much inbreeding, partly because the number of loci contributing in any cross between two parents will become small because you've lost most of the variation. So you can't push this to very high levels of F. In terms of including dominance, it's also tricky because you do need to include directional dominance and inbreeding depression and so on, so you have more quantities that you need to track. So certainly the simple infinitesimal model doesn't work with dominance. On the other hand, I still think that the core result, that selection does not perturb things very much in the limit of large numbers of loci, will hold, but we haven't worked out the details. This December, Alison is coming to visit, and Amandine, so we'll sort it all out then. We said that last December as well. Yes.
Any other questions?
I was wondering if you could expand on your comment about the omnigenic model. Jonathan Pritchard put a paper on bioarchive 2 weeks ago. I don't know if you've seen that one. Yeah. And so the model, I think originally the core genes was a few core genes, and he was advocating that one should identify those genes. Now the model is saying that it's hundreds of core genes. Does that omnigenic model add anything onto the infinitesimal model?
Yeah, I was just putting that in really just to stir things up a bit. Because,
well, that's why I thought I'd ask the question.
Exactly. So, but I think the infinitesimal model works even if, of course, the underlying genes are influencing particular pathways. And, you know, it could be, in principle, each trait has its own, you know, set of genes which are largely non-overlapping. I don't think that's the way it is, but that's a separate question from where the relationship between pathways and traits. So the infinitesimal model simply requires that there's a lot of things affecting each trait. The big puzzle to me, actually, which I really don't understand, is how we define traits and how many traits there are. Because, of course, if there are an infinity of traits, you need an infinity of genes, and the whole thing breaks down. They must overlap simply because of the number of traits we have.
Yeah, there was a paper by Martin and Lenormand who tried to estimate the number of traits. I can't quite remember the details, but they came up with something like 4, which seemed a little low to me.
I'm sure I have more than 4 traits. I'm a more complex organism.
Any other questions? Well, that's exemplary. We're 5 minutes ahead of schedule. So, if anybody wants to take another pot shot at Nick there. Okay, well, thank you very much. Can people hear at the back with just standing by the... You have any problems, are you? Okay, let's try...